My name is Tara Newman, and I am from the British Columbia Centre for Disease Control and the Public Health Agency of Canada National Microbiology Laboratory, and I'll be discussing our validation of the tool TBProfiler for the prediction of antimicrobial resistance genes in Mycobacterium tuberculosis complex at the BCCDC. The BCCDC is located in Vancouver, British Columbia. In summer of 2023, the BCCDC TB Mycobacteriology Lab became the first provincial laboratory in Canada to transition to universal prospective whole genome sequencing-based analysis for M. tuberculosis, or MTB, genotyping. This approach allows for concurrent genotyping, identification of drug resistance markers, and identification of MTB complex subspecies. Phenotypic drug susceptibility testing serves as the current gold standard for determining antimicrobial resistance, or AMR, in MTB complex. However, the prolonged turnaround time may delay treatment of tuberculosis patients. Whole genome sequencing is an alternative method allowing for rapid detection of mutations associated with AMR. TBProfiler, developed by Jody Phelan, provides AMR predictions by comparing genetic variants of sequenced isolates to its drug resistance database. To validate TBProfiler for use at the BCCDC Public Health Lab, genotypic predictions were compared to phenotypic drug susceptibility testing data for various antibiotics including isoniazid, rifampicin, and fluoroquinolones. Isolates were classified based on concordance between phenotypic and genotypic results. Phenotypic data was collected for approximately 2,000 isolates tested at the BCCDC and the National Microbiology Laboratory for resistance to isoniazid, rifampicin, fluoroquinolones, and a number of other antibiotics. Each bar represents the different antibiotics and shows the number of resistant and susceptible isolates. Isolates resistant to isoniazid at a critical concentration of 0.1 micrograms per ml were further tested at a critical concentration of 0.4 micrograms per ml. And isoniazid was the only antibiotic tested at multiple concentrations. TBProfiler was used to generate AMR resistance predictions for the same isolates, and the genotypic and phenotypic results were compared and assessed for accuracy, sensitivity, specificity, and clinical validity. The positive predictive value, or PPV, of a mutation refers to the probability of a mutation to correctly predict resistance. The PPVs of the predicted mutations from the World Health Organization Catalog of Mutations in MTB complex was used to categorize the TBProfiler genotypic results by high, greater than 90%, medium, 75 to 90%, low, less than 75%, and not available based on the WHO PPV solo, which is the PPV of the mutation conditional on being solo, aka not in combination with any other mutation. On the x-axis here are the resistance mutations we detected in our validation dataset, and on the right is the antibiotic these mutations confer resistance to. In this case, we have rifampicin. The graph is faceted by the WHO PPV categories from low to high, left to right, and on the y-axis is the number of isolates that had this mutation, and they're colored blue if they are true positive, meaning the isolate has the resistance mutation and was known to be phenotypically resistant, or they're colored red if they are false positive, meaning they have the mutation but are known to be phenotypically sensitive. Categorizing the results by high, medium, and low WHO PPV showed a clear trend of high PPV mutations having a greater number of true positive predictions for rifampicin and isoniazid. Discrepancies were observed between phenotypic and genotypic data, particularly for mutations associated with low-level isoniazid resistance. On the top of this graph is isoniazid resistance at the lower 0.1 concentration, and the bottom is the higher 0.4 concentration. This INHA and this FABG1 mutation appear to confer low-level resistance to isoniazid, meaning isolates with these mutations may be resistant at lower concentrations and susceptible at higher concentrations. The same trend was not observed for fluoroquinolones, where mutations with a low PPV were predominantly concordant with phenotypic data in our dataset. So from our validation data, we determined the validation status of the individual mutations, which is appended to the mutations in the TBProfiler database, along with the WHO PPV, so this data is output with each TBProfiler prediction to aid in result reporting. So in conclusion, the comparison between phenotypic and genotypic AMR data is highly dependent on the antibiotic, sample size, and specific mutations present within the validation dataset and database, which can be significantly influenced by the local TB epidemiology. The lack of mutation is insufficient to rule out antibiotic resistance. Phenotypic testing remains the gold standard for determination of antimicrobial resistance. Cases where a WHO-classified resistance-predicting mutation is detected in conjunction with a susceptible phenotypic result should undergo dedicated review by the microbiology laboratory and the clinical team. TBProfiler was validated at the BCCDC Public Health Lab as a predictive assay. Continued validation of new isolates' phenotypic drug susceptibility and mutations is required by clinical laboratories for optimized and reliable clinical application. I'd like to acknowledge everyone on these teams listed here that I'm fortunate enough to work with. These are the links to the TBProfiler GitHub pages. Please feel free to contact me with any questions you may have, and thank you for listening.